Yeah, so we'll start with that more specific discussion on this in vivo biocompatibility assessment with reference to the following points, like selection of animal model, selection of the defect model, and design of a study as far together with the qualitative and quantitative determination of biocompatibility. Now, if you recall the biocompatibility definition, this is the ability of a material to perform with an appropriate host response in a specific application, and this has been mentioned time and again earlier that biocompatibility is a application specific property. And in general, it has been widely understood that biomaterials must be biochemically compatible with the host tissue. It should be non-toxic and non-irritable, non-allergenic and non-carcinogenic, and biomechanically compatible with the surrounding tissue. That is very important. And here one of the things that I have mentioned earlier, that is the elastic modulus property is important, and because elastic modulus, if there is a large difference in the elastic modulus between the biomaterial and the surrounding tissue, then that can cause aseptic loosening. And overall there is a general consensus that bioadhesive contact must be established between the materials and living tissue. Okay. What are the different animal models that are what are the different animal models that are currently used for different implant for different in vivo study. So, this table actually summarizes some of the application specific animal models selection. In the case of cardiovascular applications like heart valve or vascular grafts or cardiovascular stents like made of nickel titanium or ventricular assist devices or artificial hearts and so on, the largely people prefer, or it is reported in scientific literature that researchers used either pig model or sheep model. So, this pig model or sheep model, these are the most widely used animal models for cardiovascular applications. In orthopedic or bone replacement applications, for bone regeneration or substitutes, total hip joint or knee joint surgery, vertebral implants, craniofacial implants or cartilage applications, different models people have used, but largely it is reported rabbit model, and you can see these rabbit models are used more extensively and reported in the literature. The next level of model after the rabbit, if the material is found to be biocompatible. So, one of the things that people use more often, then they validate the biocompatibility testing of the lower animal in the higher animal, and for that people can use goat model or dog model. So, goat and dog model is the next level of model that people use. For toxicity assessments of the biomaterials, for example, in the total hip and knee joints, your wire debris particles are generated. Now, these wire debris particles, when they are transported to different parts of the osseous structure, they can cause toxicity to the neighboring tissue. Now, for the toxicity of the small nanoparticle eluates, then people use the smaller animals like rat model or mouse model. So, this is essentially used for the toxicity in vivo. Okay. So, you can use different experiments for toxicity, cytotoxicity property in vitro. Similarly, toxicity property also can be assessed in vivo. Third level of application that has been mentioned or summarized in this table is neurological. Now, neurological, there are two type of implantation experiments people have done. For example, one can use the conduit nerve conduit for peripheral nerve regeneration applications. So, there even small animals like rat or cat model is useful. People use the deep brain stimulation surgery. Now, in the deep brain stimulation experiments people can use non-human primate like monkeys, for example. So, monkeys are more used in the discipline of neurosciences and so on, and they use for deep brain stimulation or other kind of neurological stimulation experiments and see the response of the animals to, or response of the primates to their stimulation. The fourth one is ophthalmological applications like contact lens, PMMA for example, or intraocular lens. So, there people use mostly rabbit model. So, overall if you see that smaller animal models like mouse and rat, they are used more for toxicity in vivo study, but for the cardiovascular applications people use largely, or people prefer, or researchers prefer to use large animal models like pig and sheep. And for orthopedic or bone regeneration mostly the rabbit, and in higher level animal dog and goat model also is used for orthopedic applications. Neurological, it is again rat model, and non-human primates like monkeys is used. For ophthalmological applications like eye related applications, the contact lens and so on, there you can use for the rabbit model. Now, there are several standards, like one of the standards for biocompatibility assessment is that ISO, International Standard Organization, and ISO 10993 and part 6, that is essentially related to the implant effects and standards in biomaterial testing. So, this essentially tells you the most direct means of evaluating implant materials effect on surrounding tissue, and different animals are to be used, and they can be cut to size, sterilized implants and implanted, and X-ray radiography can be used as an immediate assessment technique just to see that how there is local changes around the implant site at the macroscopic scale. Now, there are several there are two important two type of response, that one is called acute response and one is called chronic response. So, acute response generally recorded, this is a part of the systemic effects. Acute response typically is recorded within 24 hours. Chronic response chronic response in any animal model can be recorded in a time scale of more than 90 days or more than 10% of animals lifespan. Subacute is typically observed within 14 to 28 days, that means 2 to 4 weeks, and subchronic it is up to 90 days, like 3 months time, or less than 10% of animals lifespan. Okay. Now, there are two types of material or tissue interaction which are important. One interaction is to create an inert surface, not allowing the adsorption of proteins and adhesions of cells, thus preventing activation of the immune system. And then examples of these types of material or tissue interaction is that heads and cups of joint processes, intraocular lenses, or blood contacting devices. So, in those cases they essentially the interaction with the host tissue essentially essentially does not facilitate any biological tissue formation or very strong biological bond formation at the material interface. Another type of interaction, it is more general or advanced strategy that aims at creation of materials which promotes the attachment, migration, proliferation and differentiation, and long term viability and cell functioning in a controllable manner. And this is important for the bone regeneration purpose like bone implant and other tissue replacements. So, one of the popular example for that is 45S5 bioglass also. So, this is also one of the important material that which has been discovered by Professor Larry Henge quite few decades ago. So, 45% silica, it has 45% silica, that is S5, and there is certain ratio of phosphate P2O5 and Na2O also in this kind of glass materials. So, other things are important, that in terms of that animal selection is that why certain animals are preferred over lower order animals. Typically the one general principle that is observed in the preclinical testing is that if useful or if a small animal model can serve your purpose, you should necessarily use the small animal model, and one should necessarily use small animal model over a large animal model. That is number 1 point. Number 2 point is that if small animal model gives you satisfactory biocompatibility response, then one has to validate that similar biocompatibility response, or one has to validate the biocompatibility response of the same animal in the larger animal study. For example, if some animal when implanted in the rat model or mouse model gives good biocompatibility property, the next level of experiment should involve higher animal like rabbit or goat or sheep model before one can even think for that going for clinical trials in human patients. Second, the third consideration is that whenever you use certain animal model and you have to place certain material at certain anatomical location, how easy to access that particular organs in that specific animals, that needs to be considered as well. For example, if you want to use for cardiovascular applications, the first primary consideration should be whether how easily you can access that animal's heart, so that you can put some cardiac patch, or you can do during the surgery you can place some artificial heart valves in that particular animals. So if the animal is a very small animal, then often it is very difficult to access that particular organ in that particular animal. So that accessibility to that organ in questions or organ in terms of that particular specific application is also equally important. Fourth consideration, and perhaps more scientific consideration, is that how closely that animal bone structure, bone microstructure, bone macrostructure, and bone properties or bone remodeling properties can match with that of that human system. For example, if you take some rabbit's femur, whether rabbit's femur, their load bearing capability, their bone remodeling properties, everything, whether how close it is close to that of that human femurs. So these kind of questions needs to be critically thought or needs to be critically answered while designing the animal experiments. Okay. More discussion on the biological response of the implant. So this is your left hand side, this is that implant materials which is placed in certain anatomical location of an animal, and this is your time t is equal to 0, okay, and this is that x axis is the time scale, and this time you can consider it is in terms of days, because most of the biological events that is taking place at the implant bone interface, that is essentially kinetically quite slow process. Now at the interface, what you can see, that is a neutrophil activity. Now just few in the last module itself, I have discussed about the blood level compatibility of synthetic animals, blood level compatible synthetic implants. So therefore again blood compatibility takes an important role, simply because as soon as the material is implanted in the material in any animal system or in any living system, different blood elements, they come and interact with that material interface first. So this is the neutrophils, you can see there, and there is also monocytes that has been marked here. So these proteins are absorbed on a biomaterial substrate, and this has been this is one of the precursor for further cell attachment to the biomaterial substrate. So this protein absorption takes place within few hours of that implantation. Then after that you have fibroblast cells, because fibroblast cells is that connective tissue cells, it forms this connective tissue. These fibroblast cells, they come and adhere on the material surface, and that is the reason why fibroblast cells are used more widely and more extensively in screening biomaterials at the in vitro level. I repeat, fibroblast cells is one of the widely used cell line which researchers use while assessing that in vitro cytocompatibility property of any biomaterial, because during wound healing process and during various implantation in that animal model, fibroblast cells are known to interact with the materials as the first cell line which interacts with that synthetic materials. So therefore it is important to see how a material supports the growth and proliferation of the fibroblast cells. Okay, then comes the matrix deposition. Now in the matrix deposition means once fibroblast cells, which you can see there is a very characteristic elongated kind of morphology or star shaped morphology, mononucleate cell, and then once fibroblast cells then adhere, then what happens? Then they also secrete some protein molecules and some biological macromolecules, and that will help to synthesize extracellular matrix. And this extracellular matrix, you can see essentially these collagen fibers and elastin, and these collagen fibers, they are dispersed in the matrix, and these collagen fibers provide kind of mechanical support to these or structural support to the fibroblast cells. There is also other things that has been shown in this slide is that macrophage activity, that is also equally important. And then towards the later term of the implantation or post implantation, you also see multinucleated foreign body giant cells. FBGC stands for foreign body giant cells. So, this is the multinucleated foreign body giant cells, they also become activated. Now, this entire process you can see right from the t is equal to 0 to t is equal to 3 to 4 weeks time period, up to 3 to 4 weeks, this entire process takes place. And what is the net result? Net result is that there is something called fibrous encapsulation. So, that means all these biological components, this fibroblast, monocytes, extracellular matrix, neutrophils, they kind of make kind of a shielding around the biomaterial interface and at the biomaterial surface, and as a result this biomaterial itself, which is marked as a star, will not have any direct biological interaction with rest of the tissues or with rest of the components of the living system where it is implanted. Instead, this biomaterial is now shielded from the rest of the osseous system with the formation of this fibrous capsule on the material surface. Now, after this showing this biomaterial, the various biological events at the material substrate, the next thing that I have shown you that you know that within the 2 weeks of time frame up to 12 to 14 days, that what are the different stages that is taking place. One is the inflammation and second one is the repair. During inflammation and repair, how the different cells or proteins, they increase in their numbers. So, if you start with the neutrophil activity, it certainly increases, this is in that arbitrary unit, and then it goes saturated and then decreases. Then if you see monocytes, although they increase in number, their numbers is much less than that of the neutrophils during the inflammation process. And I have mentioned in the last module also that inflammation is one of the biological events which one cannot avoid irrespective of what kind of material that you put in, irrespective of whether you are putting it simply by well aseptic conditions or during what kind of surgery, that inflammation is bound to happen whenever any synthetic non-living material is being placed in an otherwise living system. Third one is the fibroblast activity, or fibroblastic activity goes to a peak almost like a near to 1 week time frame. Now, collagen synthesis also starts off from once this sufficient number of fibroblast, and you can see there is certain kind of a peak value of fibroblast, and there the collagen synthesis also equally picked up, and it increases further during that during the post implantation period. Blood capillaries formation also one starts in this neutrophils activity and other things. The blood capillaries also keeps on increasing, picking up, and that increases up to 1 week post implantation. So, once this inflammation period is over, then repair stage, fibrous activity goes slowly down, but collagen activity is recently picked up, and that helps in the tissue repair. So, variation depends, this temporal variation of these different acute inflammatory response and repair response depends on the extent of injury created during implantation and the size, shape, topography, chemical and physical properties of the biomaterials. So, that means that what is the size of the biomaterials, like whether it is a segmental kind of defect or whether it is a very small defect, what kind of surgery has been done during the implantation, what is the surface topography, whether it is a smooth surface or whether it is a very rough surface. So, all those properties will determine what is the temporal variation which I have explained to you in last 5 minutes or so. Okay. So, having said this, that all this cellular activity around the biomedical implants, or what is the tissue response to these implants, biomedical implants, that needs to be carefully assessed using certain microscope, which can enable you to identify different cell types in the tissues in the neighborhood of the synthetic materials. Now, this is possible only with the histological analysis. So, histology means that is you are trying to assess the structure of the tissues and the cells that contained in it. Now, this is a typical example of a long bone structure, you can see. So, in the long bone structure, I have put some kind of holes here. So, this is the defects essentially you are creating. This is called femoral defects. So, you take rabbit's femur, then you make these 3 holes. Now, typically the diameter of this hole is 2 millimeter, okay, according to the ISO standards. The length of the implant that you can put here is somewhere around 5 to 6 millimeter in length. Okay. This is the typical International Standard Organization specified guidelines for the in vivo biocompatibility in the rabbit model. Now, once you make it whole, insert it, and then you can do these experiments. You can close the wound, and then you can allow the rabbit to live for certain period of time, like 1 week, 4 weeks, and 12 weeks, and so on. Then after that, you can do series of dehydration in alcohol in the explants, what we call. This is implants. Then it is taken off. Then you do all the kind of chemical treatments. You embed it into polymethyl methacrylate resin. Then you embed it in bone, then you make the thin sections of the bone, and thin sections you can polish it in a diamond paste on a polishing wheel. Then after that you can stain the histology sections. Now, this staining can be done using different histological stains depending on what part of the tissue structure you want to reveal, or in other words which cells in the tissues that you want to reveal. One of the staining agent that is quite commonly used is the Stevenel's blue staining, and then after that you can do optical microscopy observations. So, essentially first you take the explants, you fix it, you dehydrate, you clean it, then you embed it in polymethyl methacrylate, then you cut the sections, and then after that you do this staining and observations. Now, this is a typical histological stains, and these histological stains can be used depending on what kind of tissues that you want to see it. That one of the commonly used histological stain is hematoxylin and eosin, and second one is that Masson's trichrome stain. This is an alternative stain. Now, what are the things that you want to see more clearly? This is the blood vessels here, and this is also epithelium. You can clearly see the cytoplasm is stained pink in most of the cell type. This is a cytoplasm in the stain, and nuclei are stained purple, and this cartilage also is stained purple. In the Masson's trichrome staining of the same trachea, which you have seen here, the same things that we are trying to see here, muscle stains brown, and also in epithelium is stained green in this Masson's trichrome stain. Other things you can see is that collagen also, and collagen in cartilage and connective tissue is stained green. So, these two stains are more widely used. This is again one of the hematoxylin and eosin stained image in 40 times magnifications. This is taken from Color Atlas of Tissue Response to Biomaterials, which is written by Meera Mohanty and sub-reference group from Sri Chitta Institute of Medical Science and Technology. So, we have taken this section from their book, and then you can see that how this inflammatory response, that is which is a normal physiological reaction of a vascularized living tissue to trauma, and essentially it facilitates the invasion of the foreign substance. And you can see that what are the different cells that which are participating in this inflammatory response. The other thing is equally important to understand is the foreign body response. So, this foreign body response is indicated by the presence of what I have told you before, foreign body giant cells. This foreign body giant cells is essentially multinucleated cells and components of the granulation tissue like macrophages, like fibroblasts, or capillaries in varying amount. So, macrophages, fibroblast means that is the connective tissue cells which and blood capillaries in varying amounts also should be present. And these entire things again you can study from the tissue sections, and tissue sections needs to be stained by hematoxylin and eosin, this is the staining agent. Now, if you are from material science background, this staining can be better realized as equivalent to etchant what people use in the microscopy sample preparation for material science. So, etchant is a chemical etchant. For examples, if you use titanium material or titanium 6 aluminum 4 vanadium material, this material once you need to try to find out that what is the grain size of the material, you use certain reagents called Kroll's reagent. So, let me not get into more details in the what is the composition of the Kroll's reagent and all, but for the time being you realize that Kroll's reagent is a chemical solution. This chemical solution etch the material, and differential etching at the grain boundary region and around the grain will make you distinguish the grain boundaries from the rest of the material surfaces. So, essentially you can see very nicely hexagonal grains or whatever shape of the grains that you can find in the material, and this grain boundary region is etched differently than rest of the grain bodies by whatever reagents you are using. Now, taking these concepts back to that kind of histological analysis, you can see that whether you are using hematoxylin eosin or Mason's trichome, this is also certain reagents, and that kind of helps you to identify different tissue components here. Now, tissue components are certainly very different type of structures altogether from a general material, whether it is a titanium, copper or stainless steel or so on. So, compared to that, it is a material, you do not see grain structure like this. However, you do see different kind of cells which have a different morphology in the tissue, and these particular reagents help to distinguish the different cells which are contained or different cellular features which are contained in their tissue structure. Okay. So, I hope I have been able to kind of make you understand that the use of different staining agents in the histology sections, and different cellular morphology they would appear in different features, and that will help you to understand that what kind of cellular activity is there in that particular tissue response. The final stage of the foreign body response and the healing process is the development of fibrous encapsulation, and this fibrous encapsulation process, I think I have spent sufficient time in this particular slide to explain you what it meant by fibrous encapsulation and what are the events that finally lead to the to this particular final biological event leading to fibrous encapsulation. Now, this fibrous encapsulation also involves, you know, that activity towards the end stage of the fibrous encapsulation is the foreign body giant cells, and that has been mentioned here that fibrosis and fibrous encapsulation, this is two separate processes. One is the replacement of tissue by parenchymal cells of the same type, and second one is the replacement by connective tissue that constitute the fibrous capsule itself. So, these independent processes, they are controlled by the growth capacity of the cells and tissue receiving the implant and the persistence of the tissue framework, and also to some extent on the degree of injury. Now, this has been mentioned before that depending on what is the kind of surgery that you are using, depending on how severely that wound has taken place when you are putting the material into it, this fibrosis and fibrous encapsulation process taken place in different extent. So, this is again last example, this is again hematoxylin and eosin stained image, which is again taken from Meera Mohanty's book on the color atlas of tissue response to biomaterials, and you can see this tissue response, you can see that here how different cells are well aligned in this, this is the sections and on which you have used the stains, and this fibrosis encapsulation can be confirmed from histological analysis itself. So, I think I would stop now, and so next I will continue very briefly on this tissue response again, and after that we will go to the different topics as part of this course.